In the name of God, hi dears. Our topic for today is about the muscle tissue. That is another type of main tissue in human body. In this session, we should know about the muscle tissue characterization, different type of muscle tissue, and microscopic structures of different type of muscle tissue as skeletal muscle, cardiac muscle, and smooth muscle. Muscular tissue is the main tissue of the heart, skeletal muscles, and walls of the hollow organs, and it's make up nearly half the body's mass. There are three types of muscle tissue in the human: skeletal muscle tissue that packaged into skeletal or striated muscle make up the 40% of body weight. Second type is cardiac muscle tissue that located only in the walls of the heart. And the third type is smooth muscle tissue that occupies the walls of the hollow organs such as stomach, small intestine, and others. Here you can see different type of skeletal muscle under light microscope with a schematic picture this side and a special staining in this side. Skeletal muscle or striated muscle because of the presence of the striation under light microscope named striated muscle, cardiac muscle, and smooth muscle. Muscle tissue have different function in the body that the main important of them is a movement, such as skeletal muscle that moves the body by moving the bones, and smooth muscle that move the wall of the hollow cavity. Squeeze fluids and other substance through hollow organs, that is a function of the smooth muscle that forms some sphincter in different organs. Maintenance of the posture that enable the body to remain sitting or standing, joint stabilization, heat production, allow breathing to occur, that the diaphragm muscle or other muscles of the respiratory system, support soft tissue such as the muscles of the abdominal wall, guard body opening, entrance and exit such as sphincters, and store nutrient reserves. At the first, I want to explain about the skeletal muscle or striated muscle. Skeletal muscle named because most of them attached to the bones, because of it named skeletal muscle, and also because under microscope they have striation named striated muscle. This muscle tissue include long, cylindrical, and multinucleated cells, and its function is voluntary movement of the body, locomotion in the eye, and manipulation of the environment, and facial expression and voluntary control. Located in the skeletal muscles that attach to the bone, or some of them such as facial expression muscle occasionally attached to the skin. Muscle tissue, as same as to other tissue, include the cells and also ECM, but the ECM amount is a few amount. But the characterization of the muscle cell are similar to other cell. They also have plasma membrane, cytoplasm, and nucleus. But the name of these structures different in the skeletal muscle cell. Plasma membrane of the muscle cell called the sarcolemma. Sarco means meat, lemma means membrane, and the sarcolemma means the membrane of the muscle tissue. And the cytoplasm of this tissue also called the sarcoplasm, that include glycogen that used for synthesis of the ATP The glycogen is a source of energy in the muscle tissue, and the cytoplasm of the muscle cell include the red-colored protein called myoglobin. Myo means muscle, and globin is a type of protein that have red color. That this myoglobin give the red color to muscle and also binds to oxygen molecule. And also the smooth endoplasmic reticulum in the muscle cell have a specialized function. I think maybe you know it, that store the calcium ion, and its name also is different to other tissue and named sarcoplasmic reticulum. But the microscopic structures of the SER and also cytoplasm and cell membrane is similar to anything that you know about them in other tissue and also in the cell section. Skeletal muscle fibers are equal to the skeletal muscle cells because the cells of the muscle tissue is very long. Because of it, the scientists named them to fibers. They developed from fusion of mesodermal cells that named myoblasts. These myoblasts in differentiation to skeletal muscle, some of them fuse together to form the skeletal muscle fiber or skeletal muscle cell, as you see here. Most of the myoblasts, maybe around between the 100 to 150 of them, fuse together and form just one skeletal cell or just one skeletal muscle fiber. Because of the fusion of the high amount of the cell, yes, they are multinucleate cell. These cells, the myoblasts, fuse together and formed a very long cells that named muscle fiber or cell. And after that, the contractile protein synthesis occurred in their cytoplasm. Some of the myoblasts in the embryonic period not fuse together and not differentiate to muscle fiber. They remain as a stem cell or undifferentiated cell in muscle tissue that named satellite cell. That this cell act as a stem cell, can do mitosis division and differentiate to myoblast and muscle fiber. They act as a stem cell. A skeletal muscle fiber or cell are very long cells, become very large, and there are some unfused cells that are satellite cells, that these cells act as a stem cell and assist in repair after injury. The arrangement of the contractile protein in the muscle tissue formed a stripped or striated pattern within myofibrils. That there are two bands in the cell, alternating dark bands that include thick filaments, and the physiologists mostly name them A bands or anisotropic bands, and the light that include thin filaments and also named I bands or isotropic bands that I explain for you by detail more. This picture shows the skeletal muscle under light microscope transfer section, longitudinal section. As you see here, the cells have striation, multinucleated, that most of these nucleus located under the sarcolemma. They located peripherally of the cell. The striation of the muscle tissue observed in longitudinal section, not in transverse section. And this part show the electron microscope of the skeletal muscle, thin filament or dark band or I band, and light band or I band that include the thin filament. And here the special staining that you can see striation, dark and light band, the nucleus that most of them are flat and located in the peripheral part. The functional and structural unit of the skeletal muscle cells named sarcomere. Sarco means meat, mere means unit. That in each sarcomere that located between two Z disc from here, Z disc, until here, another Z disc, this structure named sarcomere, that in each cell or fiber of the muscle, skeletal muscle fiber, there are repeated of them, maybe around 1 million of these sarcomere located just in one cell or fiber. Z disc, that also named Z line, separate one sarcomere from the next. One sarcomere to other one. Other one. Yes, it's surrounded, it's limited, one sarcomere. Each sarcomere includes two types of filaments. Thin filaments or actin filaments that located toward from the Z disc toward the center of the sarcomere. These are our thin filaments that include the actin filaments that joined or attached to Z disc by some structural protein, and they are toward to the center of the sarcomere. And thick filament that include myosin filaments that located in the center of the sarcomere, this yellow color. In each sarcomere that located between of two Z discs, there are two types of filaments, thin filaments and thick filaments. A band, as you see here, is full length of the thick filaments. Full length of the thick filaments, also named the A band, that mostly used in the physiology. In the histology, mostly we use the thick and thin filaments. I band, region with only thin filament, just from here. Yes, just include the thin filaments that the Z disc passes through the center of each band. Yes, the I band is shared between two adjacent sarcomere that include just thin filaments, and the Z disc bisect the I band. The H zone located in the center part of the sarcomere that include just thick filament, no thin filament. This part is H zone, and in the middle of the H zone and also A band, there is a line that is a midline. It's a midline of sarcomere that contain myomesin and also creatine kinase. There are some supporting or regulatory or structural proteins that hold the thick filaments and also thin filaments to different parts. For example, the myomesin is a supporting protein that holds thick filaments together to the edge zone here. And there are other type of structural protein that I explained for you one by one. And you can see here another one that is a titin that hold the thick filaments to the Z disc. The supporting or structural proteins, most of them are for the bonding or holding of the thin and thick filaments to other structure, and also maintain the diameter and the length of them in the normal length. A thick filament contains twisted myosin subunit. Yes, twisted myosin subunit. That each of these molecules include the tail or the light chain or the heavy chain tail that binds to other myosin molecule, as you see here. Yes, a high amount of the myosin molecules twisted together, and all of them attached to the M line. And the head that made of the two globular protein subunit that located near to the thin filament and react to them. The titin strand recoil the myosin filament after stretching them, and in the head of the myosin there is the ATPase enzyme that convert ATP to energy of the motion. Thin myofilaments composed of three major proteins: F or fibrous actin, tropomyosin, and troponin. The fibrous actin include two strands of fibrous form a double helix of them. In the body, in the all of the cell, there are the actin filament under the cell membrane that form the terminal web, and they maintain the shape of the cell. In the cell, the actin have monomeric structures, as you see here. Monomeric or globular structure also in the muscle cell. But when they want to involve to formation of the sarcomere, they should change to fibrillar or fibrous actin. Yes, as you see here. They form the fibrous actin and twisted double helix of them. The joining of the globular or monomeric actin to formation of the fibrous actin done by tropomyosin, that is a double peptide chain that have some site for attachment of the globular actin, as you see here. Yes, this is the number of the globular actin that the tropomyosin joined or bind to them and form the fibrous or fibrillary actin. In the last slide, yes, write that composed of G-actin monomer, each of them that have myosin binding site, and also the tropomyosin binding site. Tropomyosin is an elongated double helix protein, binds along the groove of the F-actin that form the F-actin or fibrillary actin, and troponin composed three subunits: troponin A or troponin I, troponin actin or troponin inhibitor that binds to actin or inhibit the binding of actin to myosin. TNT binds to tropomyosin and TNC binds to calcium ion. These three components located in thin myofilament that the thin filaments, actin filament, act as a contractile protein, and tropomyosin and troponin are supporting or structural protein. As I said for you in the last slide, the actin in the normal condition in the cell have actin monomers, but in the sarcomere they should form the actin myofilaments that they join together, bind together by binding to tropomyosin double chain peptide and form the fibrillary actin. Also, each of these monomeric structures have myosin binding part that in the contraction procedure, the myosin bind to this actin. And troponin have three subunits. Troponin I that bind to the actin, troponin C that bind to the calcium, and troponin T that bind to the tropomyosin. This slide shows the thick and thin myofilament location in the sarcomere. As you see here, around maybe 100 of the myosin molecules in the tail twisted together and joined to the M line by myomesin, and also the myomesin hold them together in H zone. And the head of them is toward to the side of the sarcomere. They are located near and close to the actin, and the actin have the myosin binding site that in the contraction, the head of the myosin attached to the actin myofilament. And this is a thin myofilaments, include the thin actin filaments, tropomyosin and troponin. And here show the different part session in different part. As you see here, for example, in the M line and the H zone we include just thin filaments, but in the A band we can find the thin and thick filament, both of them. And also in the I band include just the thin filaments, and we show the thin filaments that attach the myosin to the Z disc. In the contraction, sliding filaments occur between the filaments. The thin filaments of the sarcomere slide toward the M line between thick filaments. The overlapping, as you see here in the contraction, the overlap between of the I and A band increased, and the length of the sarcomere decreased, and finally this occurs in all of the sarcomere in each cell, and the length of the cell in the contraction decreased. That about the contraction you know more in the physiology.